Thank you. Thanks, Susan. Today I'm going to give you a brief overview of what I've been doing in the last few years and what we are planning to do in the next few years as well. Basically, our lab is interested in biological process. It's called ectodomain shedding. Ectodomain shedding basically is a process occurs on the cell membrane. About its irreversible process, it requires a protease. About 2% of membrane proteins undergo that process, so it's very specific. So basically, I'll just get my pointer here. So basically, a protease is required to cleave off another membrane protein to release the ectodomain of that protein. So that soluble ectodomain basically can act on the same cells to elicit cellular response. It acts as an agonist or an antagonist, or actually goes and work on adjacent cells or neighboring cells, or even reach the bloodstream to elicit cellular response distant from where it was cleaved, basically. So ectodomain shedding happens to a lot of substrates. It can occur on adhesion molecules, growth factors, cytokines, enzymes, you name it. And also, therefore, it's implicated in a wide range of pathological conditions, including inflammation and cancer. There is a group of proteins or proteases called adisintegrin and metalloproteinases, and this family of proteinases or proteases is basically the most studied family in terms of ectodomain shedding. We are interested in one of these proteases. It's called the ADAM17. That's the most studied protease in that family. It was originally named as TNF alpha converting enzyme because it's the first substrate for that protease was TNF alpha. So it's required to cleave off TNF alpha to activate TNF alpha, basically. So ADAM17 has more than 80 known substrates. It can cleave TNF alpha, it can cleave Notch receptor, EGFR ligands, and also interesting for us is interleukin 6 receptor. So as you can see, a wide range of substrates, and all these substrates are important in cell proliferation, inflammation, and so on. So ADAM17 is important in inflammation and cancer. So it's for us, it's a potential therapeutic target, basically for lung cancer. So ADAM17 is tightly regulated. It gets produced as an mRNA, and then the protein basically gets produced in the endoplasmic reticulum. So ADAM17 has to get attached basically to a regulator called IROM, and there's two types of IROMs, IROM1 and IROM2. And once the protein is produced and loaded on the IROM, it gets transported to the Golgi apparatus, where it undergoes a maturation step. So ADAM17 gets matured by another enzyme. It's called furin to release another or certain peptide in the catalytic site of ADAM17. It's called prodomain. And then ADAM17, the active prodomain, the active ADAM17 goes to the cell membrane to elicit its cellular response. One important note here is the ADAM17 prodomain. It's a natural inhibitor of ADAM17. It's very specific to ADAM17, and it's nicely inhibiting its biological activity. And we use the ADAM17 prodomain as a therapeutic basically in our models, which I will show you in a second. Interleukin 6 receptor is one of ADAM17 substrates, and it's really important in driving lung cancer as well. So our lab has shown that interleukin 6 receptor is important in driving a subtype of lung cancer. It's called non-small cell lung cancer. So how is IL-6 receptor working, basically? So interleukin 6 has two modes of signaling. One, it's called classical signaling, which requires interleukin 6 to bind to the co-receptor GP130 and also membrane-bound interleukin 6 receptor to elicit cellular response. There is another mode, which is the pathological mode of interleukin 6 signaling. Basically, it requires ADAM17 to cleave off the membrane IL-6 receptor to release the soluble IL-6 receptor. and then the soluble IL-6 receptor binds to interleukin-6 to make a complex, and that complex binds to the co-receptor to elicit cellular response. Usually, trans-signaling is associated with pathological conditions, including inflammation and cancer. So the logical way to inhibit trans-signaling is to inhibit the ADAM17 protease, basically, and that's what we've done in a non-small cell lung cancer. So lung cancer is the most common or the most lethal cancer worldwide. It can be classified into non-small and small cell lung cancer. Our work started on the non-small cell lung cancer because it constitutes about 85% of lung cancer patients. The most common subtype of non-small cell lung cancer is the lung adenocarcinoma, and KRAS mutations is the most common mutations in lung adenocarcinoma. As you can see here, we published two studies basically showing that ADAM17 is an important therapeutic target in non-small cell lung cancer, specifically KRAS mutated lung adenocarcinoma. And we wanted to expand that work to the other subtype of lung cancer. So small cell lung cancer constitutes about 15% of lung cancer patients. The major mutations in that cancer is P53 and RB1 mutations, and these mutations occur in 90% of patients. So it's a very universal, basically. Also, there is another mutations, MYC amplifications, that occurs in 20% of patients. Basically, it's a nasty disease. It's a really aggressive neuroendocrine disease. Chemotherapy hasn't changed in the last 30 years, basically, constituting cisplatin with etoposide. The success rate of that combination of chemotherapy it's very successful. It's effective in 60% of patients. However, about more than 90% of patients will develop resistance to that combination chemotherapy, which leads to a median survival rate of up to 12 months at best. Novel immunotherapies are quite effective in non-small cell lung cancer. However, in small cell lung cancer, they are completely ineffective. So there is always a need to discover new therapeutic targets, basically, for small cell lung cancer. So I'm going to show you a bit of our work on non-small cell lung cancer. So to study ADAM17, we use specific mice. It's called ADAM17-EX mice. So knocking down ADAM17 is lethal in mice. So we use that mice that basically has about 90% of ADAM17 in their body is knocked down. And there is about 5 to 10% of ADAM17 still expressed in these mice to keep them going. We crossed these mice with our lung cancer models. So the model for lung adenocarcinoma is KRAS G12D. We cross that model with ADAM17-EX. And you can see after activation of the mutation, you can see the area of parenchyma affected by tumor lesions in the control mice significantly reduced when we knock down ADAM17. And you can see here the area of parenchyma is significantly reduced in the EX mice, and also the number of tumors significantly reduced. We have another model for cigarette smoke carcinogen. It's called NNK, or nicotine-derived nitrosamine ketone. We inject the mice with that carcinogen, and then we look at the surface tumors on the lungs of these mice. Again, as you can see in the wild-type mice, you can see those dots of basically tumors on the surface of the lungs. But in the EX mice, you can see significant reduction in the number of tumors on the lungs of these mice. We also use the ADAM17 prodomain as a natural inhibitor of ADAM17, and we treat the KRAS G12D mice with that inhibitor, and you can see a significant reduction in tumor lesions. And we also treated a PDX highly expressing the active form of ADAM17 with that prodomain inhibitor, and you can see it significantly reduced the tumor growth of that PDX. So this project is focusing on investigating the role of ADAM17 in small cell lung cancer. So basically, we have a model of small cell lung cancer, a mouse model of small cell lung cancer. It's called the RPM. So the R stands for the RB1, P is the P53 mutation, and M is MYC amplification. So in that model, it basically recapitulates the genetic mutations we see in small cell lung cancer. So RB is mutated, P53 is mutated, and MYC is amplified in that model. And we are planning to cross this mouse model with the ADAM17-EXAX mice, which is happening at the moment. And we use proteomics to basically look at the mechanisms by which ADAM17 promotes small cell lung cancer, if it promotes small cell lung cancer. And we use a specific proteomics techniques to basically investigate which substrates are involved in ADAM17 action. And we also, we're going to use ADAM17 prodomain to treat small cell lung cancer PDX, and we see if there is an effect or not. So as a preliminary data, we treated a panel of small cell lung cancer cell lines in vitro with the ADAM17 prodomain, and you can see treating these cells with the ADAM17 prodomain significantly reduced the proliferation of all these cell lines. We have the RPM mouse model in hand. So basically, we activate the mutations using adenovirus recombinase through intranasal inhalation. And after 6 to 10 weeks, basically mice develop this nasty disease in the lung, small cell lung cancer. And also these mutations, these cancers metastasize to the liver, as you can see here, and it expresses all the neuroendocrine markers similar to human small cell lung cancer. And we can show that ADAMs are expressed or highly expressed in the lungs of RPM mice, and the active form of ADAM17 is significantly upregulated, and also regulators of ADAM17, IROMs, are highly upregulated. And also at 9 weeks of age, these mice develop a significant upregulation of soluble IL-6 receptor, which is a known substrate of ADAM17. So that's basically it. I would like to thank Cure Cancer for the generous funding, and also our lab members in the agency, and also our national and international collaborators. Thank you.